It's my great honor to welcome you all to the first panel of the 24-hour event, which is on multiscale human definitions, maps, and models. We have a great set of panelists, and it's my true honor to moderate this panel. I will set the stage a little bit because this is a big and complex topic, and I will do this from the vantage point of HubMap and CENET, which have funded much of the Human Reference Atlas work, but also as a CIFAR co-director, and CIFAR was generous enough to co-sponsor this event. This is the very first hour of 24 to come. Everything will be recorded so that you can also watch it later. I doubt many of you will be able to watch all 24 hours. In each hour, you will have a unique topic related to the multiscale mapping of the human body. And as you will see, there will be demonstrations and panels and presentations on maps and models of how we would go about mapping the human body across scales, from the whole body down to the single cell level, subcellular, to the human genome. You will also learn about funders and publishers and editors involved in this effort. And we are just super glad that you could join us on. This is the fourth event of this kind. It all started during the pandemic in 2021, continued in 2022 with the Human Reference Atlas event. We held another event last year on interactive data visualizations, and this year on the multiscale human. Some of you might be very familiar with the Human Reference Atlas. This is an effort that now engages more than 20 different consortia funded. Many of them are funded by the National Institutes of Health. Many of them are supported by the Human Cell Atlas. And in fact, this could not have been possible without the extensive networks that the Human Cell Atlas has created. The Human Reference Atlas tries to define the 3D space and shape of anatomical structures, cell types, and biomarkers of relevance to clinical decision-making. And it also then defines how new data sets can be mapped into this space, into this spatial space, but also the semantic space as it's linked to existing ontologies. And we also need to take into account demographics such as age or BMI or ethnicities. We want this atlas to be computable, so there are many anatomical atlases that are books, but there's no API to them. Here we want it to be published via APIs with many different user interfaces for different needs, but also published as link open data in an open fashion, and of course continuously evolving because technology development is vast and fast in this area of research. Again, it needs to be across scales because our human body works across scales. Standing is not falling. And so it's a very active, dynamical system that we call the human body. And it goes across 10 raised to the power of 10 scales. And it's non-trivial to create user interfaces to all this data. We recently wrote a paper together with 170 plus authors. And again, this is spanning many different consortia, and it was recently accepted and should be out beginning of next year. And in this paper, we just detail how the atlas is constructed, how researchers create these anatomical structure cell type biomarker tables, how 3D reference data and 2D reference data is mapped onto these anatomical structures, how we use AI techniques to then map experimental data, and then ultimately how we use cell type populations across the human body to connect to experimental data, but also how we use HRA-Lit, as we call it, to connect to scholarly publications. Ultimately, you want to use the atlas. You want to take a new tissue block and map it into the atlas, either because you know the spatial position or you have the cell by gene matrix or cell by protein matrix. We have OMAPs for so-called validated antibody panels to map immunohistochemistry data into the atlas. And I think spatial data, especially 3D spatial data, is especially valuable for common coordinate system constructions that Griffin will present next. And so ultimately, you can then use the atlas to predict cell by biomarker matrices if you have a location in the human body, a volume that you can identify. Or if you have cell by biomarker data, you can predict where it might have come from. And that's really the atlasing functionality. But you can also use Human Reference Atlas to see what changes as disease comes in. And you see that here for lung data. But you can also look at cell neighborhoods, and that's work by John Hickey, and he will present later in this series. So thank you so much for joining us all, and I will give it over to Griffin Weber. Griffin is an associate professor of medicine and biomedical informatics in the Department of Medicine at the Beth Israel Diakonis Medical Center and the Department of Biomedical Informatics at Harvard Medical School. And within HubMap, he has been leading the vasculature-based common coordinate framework that is very central to the Human Reference Atlas effort. So Griffin, just let me know when I should go to the next slide, and I'm doing this now.
Thank you so much. You go move to the next slide. As Katy said, I'm going to be talking about the vasculature common coordinate framework. Just want to start by thanking sponsors and organizers of this event and Katy for inviting me to present today, and list of people on this slide who've helped out with this project. The idea here is using the millions of blood vessels and blood vessel pathways through the body to be able to define the location of individual cells. A cell in your body, and there's 37 trillion of them or so, they don't have an XYZ coordinate. How do we describe where these cells are and how they interact with nearby cells? So our idea here is that the blood vessels extend to all parts of the body, and can we use it like a coordinate system to indicate where you're located? So next slide, please. So how do we even begin to think about building this map or coordinate system of all the blood vessels? So when we began, we thought about this analogy of how the multiscale map of blood vessels may be similar to a multiscale map of roads. Here on the left is a map of the US National Highway System. The thicker red lines are highways that have larger amounts of daily traffic. When I look at that map, it looks like arterial networks and blood vessel networks to me. As you zoom into an individual city, here's Boston on the right where I live, you can start to reveal smaller streets and alleyways. Interestingly, one of the main highways that crosses through the city is nicknamed the Boston Central Artery. So maybe we're on the right track here. Next slide, please. When I'm ordering a package on the internet, I don't type in the latitude and longitude coordinates of my house. I enter in the name of a street, which is next to my house, because that's how the package will get delivered. Similarly, a red blood cell is delivering oxygen and nutrients to organs by following a pathway of blood vessels to get to those locations. We have a YouTube video that illustrates this analogy. I'll step through a couple frames of it. Next slide, please. Here we're starting with a truck on the left going down a large highway, and the red blood cell is in one of the major arteries of the body heading towards an organ. Next slide, please. The truck then is pulling into an organ and then to a neighborhood to deliver the package. Here, the blood cell is now moved into a smaller arterial vessel. And then finally, both of these are heading to the next slide, please. They're final destinations of the driveway to deliver the package. And the red blood cell is now in a capillary delivering its contents over to the adjacent cell. Next slide, please. So in order to actually draw out these blood vessel pathways, we use something called a blood vasculature to organ crosswalk diagram. Structures on the left are larger blood vessels or organ structures, and then on the right are smaller and smaller structures of those organs. On the left, we have the major blood vessel superhighways, like the aorta and vena cava. Smaller arteries branch off of these and head through different organ components to smaller and smaller items so you get to the functional tissue units. There we now reach the capillaries and head back through venules and veins to the and return to the heart. So next slide, please. Here we can start overlaying different organs. So here we work with the pancreas experts to draw out the gross anatomical structure of the organs. These are the large rectangles on the left, and then the nested functional tissue unit rectangles on the right. And then we overlay the different vasculature pathways going through there. These diagrams are very helpful in visualizing the different scales of the vasculature. When you go from left to right, as Katy mentioned, you're going through orders of magnitude and different size, and it helps us understand gaps and identify locations where we may not know as much detail of the vasculature. They're showing you some increasing complexity on this. The next slide is showing the liver, and then After that is the brain. And then again, these diagrams can get quite complicated, but they're representing, again, many different components of the vasculature and the organs and crossing many different scales. And these are a way to actually visualize all that complexity and to be able to communicate with the organ experts to build out this map and ultimately feed it back into the Human Reference Atlas as a coordinate system. Thank you.
Thank you so much, Griffin. Please hold your questions for later. Next is Karen Miga. She is an associate professor of biomolecular engineering at the University of California, Santa Cruz. She's going to present on the Pangenome, which is a reference that will enable us more complete and equitable understanding of genome diversity. Thanks so much for joining us, Karen.
Thank you so much. And so next slide, please. So my talk is actually focused on something that we consider a foundational tool for a lot of biomolecular science and genetics and genomics, and that is the celebration of the first map of a human genome. And this is the A, T, Cs, and Gs that was first released back in 2001 and celebrated for decades. It's been driving a lot of innovation and new science because now we can find where genes are, much like the talk before us, like a coordinate-based system. We think about the human genome reference map, all of our chromosomes, all the list of nucleotide bases, kind of like a coordinate-based system we think about with Google Maps. No one's really looking at a precise part of your genome necessarily. They're looking at that rich annotation on top of it, what the gene is, what the variants are that tell you something about human disease. And because of all of that information we piled onto that map over 2 decades, it's become incredibly valuable for us to really begin to study human variants and their cause towards traits. However, like most of your phones or your computers or even medical devices you may have at a hospital, most technology-driven resources don't stay around for decades, like the human map has become kind of a part of our community. And so it's really due for an upgrade. We know that there are issues. There are places the map is incorrect. We know there are giant holes in the map where we're lacking millions of bases or millions of nucleotides of information. And we actually are in a great place now because technology for building maps has improved so much that we now can do this at a really high scale, not just one map, but we can make thousands of maps. We also know that using just one map isn't great for everyone who's listening to this talk, because this map may not represent the best coordinate system for you. And so by having just one map, what we're finding is it introduces biases, and this could be problematic as we start to move into a world of genomic medicine, where we're trying to custom fit drugs and custom fit different things for your particular map. Next slide, please. So we know that if you were to take a map, as I'm showing one individual, and every single human chromosome is a line, and the red parts are the parts that are really tricky. These are the complex regions of your genome that are new that our team's really specialized in bringing into focus. We know there are important regions that we can look at this map, things like HLA typing, if you're interested in organ transplant, or SMA locus, or even looking at some sites in our genome that can tell your doctor whether or not the metabolism of certain clinically used drugs is important for your map. Next slide, please. So I'm here representing a consortium called the Human Pangenome Reference Consortium. This was a call from NIH to improve that original human genome map and try to replace it with a new tool. This is something that's designed to represent global genomic diversity and all of the common alleles that we find at a high frequency. We organized a team of genomic experts. These are folks that are really good at making maps. And I've just highlighted a few teams here, like something called a T2T Consortium, Vertebrate Genome Project, as well as the HGSBC. And we've also brought in a bunch of tool makers to understand how do you actually put all of these maps together into one useful tool. Next slide, please. And so if you think about that one map before that I was showing you, now you have a series of maps, a collection, and we have to reorganize that. So instead of a kind of panel of highways, we're looking at something that looks more like a subway map. Next slide, please. In total, we're looking at about 1,000 individual maps. And we think about them as, how do you build this map? How do you then find their best alignment to one another? And then how do you build this type of subway map? In doing so, we're making everything open access to open it up to as many individuals around the world. And so this is all of our tooling ecosystems that we're hoping that scientists and non-scientists and clinicians eventually will become more familiar with and begin to integrate them more into common use. Next slide, please. You may ask who is represented. We actually have been building off of a prior consortium effort. And so we have the use of different cell lines that have been acquired through participants in different centers around the world that I'm highlighting here. Next slide, please. We've started to build tools, really highlighting members of our team here who have been using different types of sequence methods to try to build these tools together. Next slide. We have centers around the United States that are actually utilizing these different sequencing platforms in production mode. So we're actually generating this at high scale now. Next slide. So we've been celebrating a draft release of this subway map. This is something that if you're interested, you can download. I'm highlighting the key lead authors on that paper here. Next slide, please. And so what the subway map looks like, you can imagine the two roads on top. You're really trying to find all those differences between those roads, and you can model that using the subway map. You can see I'm trying to highlight for you in blue versus orange, all of the differences that you can see are between those two sequences. You begin to model them in a different structure. Next, please. So this is that focus of that part of the map that's really important for telling clinicians whether or not patient is going to be good at metabolizing drugs. And you can see that kind of subway structure with the colors that tell you where these important genes are. Next slide, please. You can begin to trace like you're going through different stations of that map, and we can begin to predict the different organization of that particular site in the genome. And that can tell someone whether or not there is more sensitivity or less sensitivity to various drugs based on what path they took through that subway system. Next slide, please. So just as a way to end this particular part of mine, I just want to say, we are just getting started. We had this initial release, and we're about to move more and more into the future, where in 2026, stay tuned, we hope to have a really big announcement of a stable release, and in 2028, we'll have our final release and celebration. Next slide, please. And so eventually what we're hoping is to replace that one map with a collection of maps that represent global diversity. And this should drive into the future basic clinical science and understanding our own human histories. Thank you again.
Thank you, Karen. I think it's very important to make this map more complete and equitable. And I'm glad to see you all working on that. All right, the next presentation is by Caterina Strambio de Castilla. I just met her recently, and I'm absolutely impressed by how much energy she brings to the table. Caterina is a faculty member at the University of Massachusetts Medical School and a Sean Zuckerberg Initiative imaging scientist. She represents work here for the 4D Nucleome network. And again, I think there are many synergies across all of these efforts, and it's wonderful to see them all presented in 1 hour.
Thank you so much. Please, next slide. Okay, so I'm actually going to present the work. So I'm involved in many different initiatives because I also very much believe that we really need to work together to learn as much as we can about the biological system and the human body in particular, and then help us with devising therapies. So, but in particular, I'm going to concentrate on my work in the 4D Nucleome. And these are, I have the honor to represent the work of many different people. And I'm going to tell you about the work that we started doing, collaboration also with Katy, to connect an intracellular common coordinate framework which we're building with the human reference atlas. Next slide, please. Okay, so the 4D Nucleome initiative is actually finishing next year. So it's at the end of 10-year incredibly productive work that we actually had our final meeting that ended yesterday. And the idea is so we learned about the genome and like a subway map of the genome. But another layer of complexity is that this genome, which is meters long, has to actually be packed in very small cells. And so what you see here is basically this capsule containing all of these like seemingly very disorganized ball of strings. But this is very important for the functioning and the activation of specific genes. And so this is what the 4D nucleome has really worked and has revolutionized the way of understanding this world. And in particular, by using two different types of techniques. One is more sequence-based, which is called, in general, it's basically looking at the bending organization of the chromatin. The other one more based on imaging. And there has been a lot of effort in integrating the imaging and the more sequencing-based information. And that's where the imaging working group and the imaging genomics working group have really attempted to give to work on. Please, next slide. So we have then worked on multiple different, because again, the 4D nucleome is just one piece and it's concentrating on the inside of the nucleus, but we really need to connect with multiscale. So we have provided leadership on several fronts, including the ability of comparing image data across different systems. So developing a metadata model for microscopy. We have developed data exchange formats for chromatin tracing, which is a brand new technique that didn't have any way of really exchanging the data in a standardized manner. And more recently, we have started working with HubMap to connect a reference atlas inside the nucleus to a reference atlas of the whole body. Next slide, please. So for image data, it's really important to be able to compare different images from different formats, from different vendors. And these are actually 3 different images of the same sample, how different they are. It's really important. Next slide. To have information about how the images were acquired, and different vendors provide very different types of information or any different different amounts of information. So we really needed to standardize this. Next slide, please. And so we brought together a large consortium of organizations from around the world to work with manufacturers to learn how to best describe these microscopes and the conditions of acquisition of images. Next slide, please. And we developed a microscopy metadata standard. Next slide, please. That we call 4D, which has brought together multiple different organizations. So 4DN, Bioimaging North America, the Open Microscopy Environment, and Quality and Reproducibility for Large Microscopy. We call this for short MDOQ, Microscopy Metadata Model. Next slide, please. We have developed tools for capturing this information in a user-friendly manner. Next slide, please. And so basically what we are doing is we are working with a lot of manufacturers to revise this model so that we will have a shared way of describing all of this information to ensure reproducibility and reuse value for image data. Next slide, please. So the next thing that we have done is work on to standardize develop standards for exchanging the results of multiplex FLISH chromatin tracing. This is a technique to be able to look at the structure of the genome by microscopy. And it's a brand new technique, so there wasn't any standards for describing this information. And these standards are very important to be able to exchange data and really learn more about how the genome is organized. Next slide, please. You can just go through this, yeah. And this is very important to actually be able to then build models. So, but another piece that is really important, so, you know, in addition to the metadata and the data exchange format, we really need to be able to compare all sorts of different types of data, not only the microscopy data, but also genomic data, and then put them in context of how they are organized in the nucleus, how they're located in the nucleus, and then put the nucleus and the cell in context of the whole body. Next slide, please. Next, please. And so the nucleus is organized in also like a little bit like neighborhoods, and the position of different pieces of the genome in relation to these different neighborhoods is really important to determine how they're going to be organized. Next slide, please. And so this is particularly important because the nuclei are very different from each other. So we need to develop a way of really mapping the position of different pieces of the genome with respect of the boundary of the nucleus or the nucleolus, like which is the structures in dark blue here, which can be very different in shape. So how do we actually compare the vicinity or the distance between different aspects of the different aspects of the genome from these landmarks? So that's why we need a nuclear coordinate framework. Next slide, please. And so we got inspired by the Human Reference Atlas, which Katy has already described. Next slide, please. To build something very similar that would allow us to map the position of chromatin structures with respect to the nuclear landmark. Next slide, please. We call this the nuclear common coordinate framework. And then we are now in the process of acquiring data to build this system because, you know, we've just started this project. Next slide, please. And so the data is being acquired at UCSD to help us develop the system. Next slide, please. And the idea is once we have a benchmarking data set to set up this system, then we can integrate it into the Human Reference Atlas so that we can connect the intracellular structure, the intranuclear structure and the chromatin organization and gene regulation with how the body is laid down so that we can ask functional questions about the regulation, the genomic regulation and transcriptional regulation of different organization inside the body. Thank you.
So it's my great pleasure to introduce Clair Walsh here. She's going to share your own slides. Clair Walsh is a senior research fellow working at the Center for Advanced Biomedical Imaging at the University College London. And together with Professor Peter Lee, she is leading the Human Organ Atlas effort. And it's great to learn more about this because this is really the opportunity to possibly at some point, if all the ethical considerations are clarified, to scan an entire human body, ideally two, one male, one female, and then to zoom in to highest resolution and really have it all in one frame of reference. And so it's super exciting, Claire, to see you here. And please do share your slides if you can.
Okay, thank you so much, Katy. Hopefully you can all see my slides.
Yes. Mm-hmm.
Yeah, so hi, thank you so much for the introduction. As Katy said, I'm Clair Walsh, and I'm here today representing the Human Organ Atlas, or the HOA, as we call it. Now, the Human Organ Atlas is based around an imaging technique, so a little bit different to what you've heard so far. Really, we're fundamentally a team that use an imaging technique called hierarchical phase contrast tomography. What you can see in the video here is an example of hierarchical phase contrast tomography in a human kidney. Now, the way this technique works is you take a human organ, like the kidney you see here. These have been donated by people who've died and donated their bodies to science. And we take that organ and we put it in a synchrotron, which I'll tell you about later. What we can then do is we can scan that entire organ intact still, and you get an overview of the kidney. So the orange that you see here, an overview of the whole entire kidney in 3 dimensions. You can then zoom in anywhere inside the kidney, and you can zoom in to the green region, and then you can zoom even further in to this kind of purpley yellow region here, which takes you all the way down to less than a micrometer voxel size, or down to the level of single cells. This data is all 3-dimensional, and these volumes are nested inside each other, and they all align with one another. Now, this technique was developed in 2021 in response to the COVID-19 pandemic to try and understand what damage COVID was doing to the lungs. But we really quickly realized that this technique was applicable across many human organs, and we quickly moved from applying it to the lung, to the kidney, brain, liver, pancreas, spleen, you name it. Now, HIP-CT, as I said, it happens at a synchrotron. Now, a synchrotron, for those who aren't aware, it's like a massive particle accelerator. You can see it here, accelerates electrons round and round this ring, and they produce X-rays. And it's these X-rays that we're using to do the imaging. Now, this particular synchrotron is called the European Synchrotron Radiation Facility. It's based out in beautiful Grenoble, France, nestled within the Alps, as you can see here. And it's quite a powerful and special synchrotron. It's the world's only 4th generation X-ray source. And essentially all this means is that there are probably about a dozen synchrotrons worldwide, but this particular synchrotron produces the most brilliant X-rays in the world. And this is really important for the HIP-CT technique. We use these incredibly brilliant X-rays to be able to produce these multi-scale images all the way from the whole organ down to the single cell. We use these 2 particular beamlines. They're called ESRF. This one here, it's called BM18. It was designed by Paul Taffero down here. And this one over here is called BM05. It was the old test beamline that we developed this on. Now, we've been working on this project, as I said, since 2021. And last year, we actually launched the Human Organ Atlas Hub. So we spent a few years just developing the technique itself. And we got to the stage where the technique is reproducible and fast enough that we could kind of open it up to the global scientific community. And we formed the Human Organ Atlas Hub, or the HOA Hub. Peter Lee here, Professor Peter Lee, is the executive co-chair, who's professor and the PI on the initial grant. But you can see here we have another 8 partners within the grant, 8 different institutions. Max Ackerman and Danny Yonick, who pioneered a lot of the COVID-19 lung work that we did. Myself, I'm the director here, Bernadette de Backer from Amsterdam, very interested in female reproductive organs and fetal development. Anastasia Yandicki from Harvard University, very focused on the brain and on understanding how we could use HIP-CT to better understand the wiring of the brain. Stein Verleden from Antwerp, another person who's very interested in the lung, and Alexander Bellier from the LADAF, who's an anatomist. And really this team represents kind of a really important mix of specialities that allow the technique to be done, but also to reach out and impact on different types of diseases, different samples, different types of analyses. Here we go. So what are we doing with HIP-CT? Well, we have kind of a few different areas. One is we're building something called the Human Organ Atlas, which you can see here, and I have a slide on in a minute. Another one is that we're doing correlation. So you've heard about from Aviv about the Human Cell Atlas, and here HIP-CT kind of sits in the middle here, and we're trying to correlate down between HIP-CT. So you can take an organ that's been scanned by HIP-CT, you can then cut it up and do histology on it, and you can then do spatial transcriptomics on that same piece of tissue. So it really gives you this opportunity to link all the way back to the three-dimensional. We're also working the other way to correlate upscale to clinical imaging, like the MR and the CT that you would have in hospital. And this is really interesting and powerful because it potentially opens a window onto us being able to understand what are kind of the microstructural things that are causing us to see certain things in clinical imaging that may be ambiguous. So if you see a smudge on a lung CT, you might not be entirely sure, is that fibrosis? Is that an early stage of a metastases? What is it? And by correlating HIP-CT up to these clinical techniques, we can start to unpick and understand that better. We're also doing a lot of quantification and modeling. Some work that we worked on with Katy and Griffin, in fact, was segmentation of the vascular system of the human kidney, going, bridging those beautiful scales that Griffin showed earlier and quantifying how many blood vessels do we actually have in the human kidney at all these scales? What is their lengths? What are their diameters? What is their tortuosity? A fourth area, which Aviv and some others have also touched on, is dynamics. The human body is not a stationary object. Now, as I said, HIP-CT relies on the organs that people have donated who've died, so they're not intrinsically moving still, but we can do what's called quasi-static motion. So you can take, for example, this is a heart, the coronary artery of a heart, and we insert a balloon catheter in, and you expand that balloon catheter while taking HIP-CT images, and it allows you to quasi-statically then reconstruct the dynamics. And the final thing, as Katy mentioned, we're hoping in 2027 to be able to scan an intact human body. To be able to do that, you have to be able to scan through both hard and soft tissue, which historically for this kind of X-ray imaging has been incredibly difficult. You can either get very good images of the hard tissue or of the soft tissue. It's very hard to get both together. And we've worked really hard to develop some new algorithms that allow us, this here is an example of a sheep's head that we image. So you can see the brain, you can see a lot of structure within the brain, and you can see all the bony tissue still as well. So the final thing I'll talk a little bit about is the Human Organ Atlas data portal itself. You can access it here. And what we're really doing is all of this wonderful data that we've collected. So we've collected well over 500 different samples now. We have put, so far, we've put 173 of our data sets up online. We have 18 different organs from 9 different donors up. And this is completely openly available to anybody in the world, scientists, member of the public, anybody can look at this data online, can download it, can interact with it. And we're really hoping that the more people that interact and use this data, the faster we'll be able to gain new insights from it. So with that, I think I hand back to Katy. Yes.
Thanks so much. And I think it's a huge challenge to make these large data sets available to people around the globe. It's actually not resolved. There was a recent Nature Methods paper coming out on the entire community getting together and arguing how to do this best, and that there's funding needed for it. And I think it's wonderful to see you go for the challenge and make that available, no matter what the cost. So thank you very much for doing that. Not trivial. All right, it's my great pleasure to introduce our next speaker, Maryann Martone. She is the Professor of Neuroscience, University of California and San Diego. And I will share this slide.
Well, Katy's getting that up. I can start on the introduction. So we have seen some absolutely spectacular work on all of the efforts that are underway to do really large-scale maps of the human body. I think the techniques that we have today allow us to map on a scale that has never before been possible. I started my career in multiscale microscopy about 30 years ago, and we could do a millimeter of tissue. We were happy. But I'm going to talk about one part that hasn't been discussed in terms of maps, and that, of course, is the nervous system, particularly the peripheral nervous system, which, of course, connects the brain to the periphery of the body. It is the part of the nervous system that innervates the end organs and is, as we know, responsible for movement and sensation. But we're also learning that it is responsible for many other things in its interactions with the central nervous system. Our understanding of how important the periphery is and the peripheral nervous system is, and these connections between the body and the brain, is the concern of a project called SPARC, S-P-A-R-C. And that stands for Stimulating the Peripheral Activity in Order to Relieve Conditions. And we're starting to understand through programs like SPARC and others that if you can manipulate the peripheral nervous system, if you can put electrodes inside of nerves and stimulate them, that you can affect all types of conditions that you would not think that you could affect. People have tried to do this for depression and epilepsy and other sorts of things that we really didn't think belonged in the periphery, but it's clear that they do. And this whole field of medicine is called neuromodulation, or you'll also hear bioelectronic medicine. And again, it is without drugs, right? This is really, you take an electrode and you use the native capability of the nervous system inside of our bodies to try to affect a whole range of human conditions. But as Katy indicated in the beginning, most of our maps of the way that the body is put together are static images that appeared in textbooks. And a lot of these have been done with gross dissection techniques where people dissected it and said, hey, can we follow where this nerve goes? And they were produced 100 years ago. And so the SPARC program was instituted by the US National Institutes of Health to deliver more detailed and integrated functional and anatomic circuit maps for this CNS end organ interaction via the peripheral nervous system. So it is a chance for us to update all of those maps that you may have seen in Gray's Anatomy and other textbooks that show how the central nervous system interacts with the periphery. And by doing this, we'll provide the scientific foundation necessary to be able to translate more effectively all of the potential promise of bioelectronic medicine or neuromodulation into effective therapies. Like many things, there's a lot of excitement around it. Things have been tried. They haven't always panned out. And so this is an attempt to make sure that we understand really what's going on. So if you go to the next slide. The SPARC portal, like the resources that you have heard about already, is a free online resource. If you go to sparc.science, you can browse and view all kinds of data sets that different researchers have collected, the 2D and 3D anatomical maps, which I will be talking about, and Peter Hunter, who is coming right after me, is actually one of the major creators of. We have computational tools, simulation platforms, and other things all around this area of bridging the body and brain. Next slide. So here in the next slide, you see actually one of these maps. So on the right, you see a human body, and on the very far right, you see the brain and spinal cord. And this looks just like some of those highway maps and subway maps that you saw before. These are the connections that come from nerve cells that sit inside of the brain and spinal cord and send their axons out to the periphery. They also send their nerve cells out in the periphery, and they send their axons back. So you may be familiar with these nerves. You may have seen maps like this before. There's something that is special about these particular diagrams. One is that these are drawn computationally. So we have a large database that has the connections of the peripheral nervous system stored. So these maps are drawn by Peter Hunter and his team through a computer program that takes that information and translates it into this map. What does that mean? It means that you can query over it. You can ask questions. You can turn things on and off. The second thing that's special about this map is that even though we think about nerves as cables that connect different parts of the body, that is 100% true, but remember those cables actually represent parts of nerve cells. So these nerve cells, if you look at any one of these highways, just like you see in a traffic pattern, things that travel along these highways may come from many different places. So many different parts of the nervous system may have cells that send their axons through these cables, and many different nerve cells that are in the periphery may send their cables back. So each one of these things is highly mixed. And our database records the locations, the detailed locations of which cells give rise to these projections, where they go, where they terminate, where is the end, or where is the synapse that communicates with either the organ, a blood vessel, or another nerve cell. So it is a very detailed database. And what that means is, if you think about it, if you stick an electrode in here, you're not just getting one population, you're getting all of those populations. And so this is helping us understand, if you take an electrode and you put it in any given location in a nerve and you stimulate it, what might all the different structures be that you will affect? And this is very important both for understanding how it is that neuromodulation therapy works, but also sometimes you get effects that you don't want. Sometimes you want to target one area, but you also get other areas. And these detailed maps are going to be what help us with this. So that was basically my brief presentation, but it is really nice to see. The nice thing is we've been working with HubMap and others so that all of our maps are aligned, and at some point we will be able to put all this information together. Thank you.
Thank you, Maryann, and thanks for contributing the peripheral nervous system table to the ASCT plus B set of tables. I think this is a really important one. All right, in the interest of time, Peter Hunter, last but not least, he comes from the Bioengineering Institute in New Zealand. And it's a true pleasure to have you here, Peter.
Thanks, Katy. Hello, everyone. I'm just going to talk, I've just got 2 slides, and I was just going to talk about one aspect that we're following up on in relation to the SPARC project, and that is using physics-based multiscale modeling. And I just wanted to talk briefly about the approach here. So what we do is if you start from number 1 on the left, we design what we call bond graph frameworks, which just make sure that the laws of physics are satisfied with models. So conservation of energy, conservation of charge, conservation of mass. And we build models that capture the properties of individual proteins. And so on the left there at the bottom of that list, you can see one particular GLUT transporter, which is a member of the SLC family. And what we find is that for a family like SLC transporters, there's about 500 genes, there's about 66 family members, but they all follow relatively similar templates. So we can create physics-based models of these proteins that can then be parameterized for individual cells and tissues in an automated way, but the underlying templates are built on a framework that ensures that the physics is satisfied. And then what we do is, in some cases, model reduction, where we can use some equations that are at equilibrium, very, very fast reactions, for example, for ligand binding or unbinding. We can simplify the models a bit. Then we build what we call number 2 there, which are functional cell units, where we automatically put together aspects of cell function from these physics-based individual protein models to cover all the different processes that go on within a cell: metabolism, action potentials, calcium handling, and so on, pH regulation. And then we again, because the models are all annotated in a way where we can then create higher level models from those functional cell units, we can create cells appropriately, create all the models that deal with the function of different cell types, such as endothelial, epithelial, etc. cell types. So what you're looking at the bottom of that number 3 are the components of a cardiac myocyte, involving electromechanics and calcium handling and signaling and myofilament mechanics. Then we put those cell types together into what we call functional tissue unit environment, where very typically you have 3D structure coming in as well. So you've got to be thinking about the organization of those cell types within a 3-dimensional spatial environment. And then we can build models that capture the function of a glomerulus in the kidney, for example, lung alveoli, and so on, often dealing with transport processes involving both cellular and paracellular transport, as shown there with that 5-layer model. And then we use those functional tissue units, FTUs, to build organ level models. And at that stage, we're almost always using what we call scaffold, 3-dimensional finite scaffolds that capture the 3-dimensional anatomy of the organ. And we can then provide the spatial variation of tissue properties across those organs with these scaffolds. And in some cases, the scaffolds are really just scaling up. I mean, in a way, a kidney is just a scaled up version of a nephron. There's often 2 types of nephron, but providing you're dealing with the spatial variation of parameters, the kidney really is a large collection of the function of a nephron. Whereas something like a heart, you very much the function only emerges at the integrative heart level. So what we do is to use partial differential equation modeling of those organs, but then use surrogate AI methods to produce much simpler surrogate models that we can plug into higher level descriptions of homeostatic mechanisms. So the final level there, number 6, just systems, are dealing with various homeostatic processes in the body, very often using AI-based surrogate models that have incorporated those lower level functional tissue units with their cells, their functional cell units and protein templates. There is a paper that describes all of this framework, which has just been published in IEEE reviews in biomedical engineering. Okay, next slide. Thanks, Katy. And then the idea is that, this is my last slide, the idea is that we're using this framework to bring in clinical data across multiple scales. So typically when you have a diagnosis of some condition, you're getting information at the genetic level, the blood biomarker level, the tissue biopsy level, clinical imaging level, and functional physiological test level, such as lung function testing, for example. And so what we're trying to do is to design these multi-scale models so that they obey the physics at every scale and across scales, but then can be linked with data sets from clinical measurements. And that means that we're in a position where we can start to construct whole body models where the interpretation of whole body data is constrained by the physics. And that means that we can use AI methods to deal with large data sets, but where those AI methods are based on physics, it's implemented through these multi-scale, typically bond graph, but also other ways of ensuring that we're capturing the physics at each scale. So thank you.
Thank you very much, Peter. And I think you have many of these scales that we all care about here on your slide. And I think it will be really wonderful to connect all these different lines of research, which we just learned about. Gary Bader won't be able to join us today. His wife was called into the hospital. She is a doctor there, so this was known that this might happen, and it has happened. But I think ultimately it is wonderful that now researchers around the globe are looking into these multi-scale human maps and models, and many of the ladies and gentlemen which presented here today are also listed here as already associated with the program, and some of you might become associated in the future, given what I just heard here. So thank you again for coming in. In the interest of time, I will end this first hour here. I would like to thank all of you one more time, and maybe we get a round of applause for everybody. And I think there are now a number of participants also which are here. And again, all of this will be shared as a recording, and it's just wonderful to see you here, and I hope we get to talk in person, ideally, maybe at the next HCA general meeting. Thank you all.